of Science Technology Belt and Road Initiative, the International Talent Program. It is our great honor to have invited Drs. Chris Sander and Augustine Lunar from Harvard Medical School. Also joining us in the panel discussion will be Dr. Yu Xue from Huazhong University of Science Technology, Dr. Rui Sun from Westlake University, and Dr. Tianan Guo, also from Westlake University. Professor Chris Sander is faculty in the Department of Systems Biology at Harvard Medical School and affiliated with the Broad Institute and the Ludwig Center at Harvard. Earlier, he was the founding department chair of the Biocomputing program at the European Molecular Biology Laboratories in Heidelberg, and with Michael Ashburner, co-founder of the research program at the European Bioinformatics Institute and director of the Z-Bio Center in the Department of Data Sciences at the Dana-Farber Cancer Institute. He was a founding member of the International Society for Computational Biology, and with Gary Stomer, founding executive editor for the journal Bioinformatics. In 2010, he was awarded the ISCB Accomplishment by a Senior Scientist Award and is currently visiting Einstein Fellow at the Berlin Institute of Health. He is one of the best-known researchers in protein structure analysis and is an internationally recognized expert in computer-assisted cancer research, as well as the founder of perturbation biology, a field that seeks to predict the responses of cells to certain disorders using computer simulations. His work seeks to assess and individually improve the efficacy of drugs in the treatment of different tumors using simulations for their treatment options down to the level of single cells. Augustine Lunar is a PhD in bioinformatics from Boston University as part of a joint program with the National Cancer Institute in the United States. He earned a Bachelor of Science in Biomedical Engineering from the Georgia Institute of Technology and is currently a staff scientist at Harvard Medical School in the Department of Systems Biology with a joint appointment at the Broad Institute at MIT/Harvard. Before his current appointments, he was a postdoctoral researcher at the Memorial Sloan Kettering Cancer Center in New York City and the Dana-Farber Cancer Institute in Boston. Augustine's research involves several areas of network biology and systematic understanding of the effects of small molecule perturbations on biological networks in cancer. In this work, Augustine has worked to further our understanding of cancer biology through the development of several mechanistic and statistical models that involve various aspects of gene regulation, genomic alterations, proteomics, and metabolism. In 2022, Augustine received the Broad Ignite grant to further his research. Over his career, Augustine has published over 35 publications, including co-authoring articles published in prestigious journals such as Nature and Cell. Augustine is also a named co-inventor on the recent patent application for a novel treatment, triple-negative breast cancer. Augustine has been a long-term member of Combine. Additionally, Augustine is a developer for several R programming language packages made accessible through the grant and Bioconductor repositories. Lastly, Augustine strongly believes in research outreach and dissemination and is a regular presenter at national and international conferences. Now, before we move on to the talks, please allow me to introduce all the panelists. Dr. Yu Xue is a professor at the College of Life Sciences and Technology, Huazhong University of Science and Technology, and the director of HuB Key Laboratory of Bioinformatics and Molecular Imaging. His research focuses on chemical modification informatics of proteins, and he has published 24 SCI papers with influence factor more than 10 in Nature Biomedical Engineering and Nature Communications. As a sole or co-corresponding author in the previous 5 years, he serves on an editorial board of many international journals, including Science Bulletin, briefings in bioinformatics, genomics, proteomics, and bioinformatics. Dr. Rui Sun received her undergraduate and master's degrees in Pharmacology from China Pharmaceutical University and her PhD degree in Biology from Westlake University, Zhejiang University Joint Training Program. During her PhD, her research interests are precise molecular classification and prognosis determination of various diseases based on proteomics. In July 2022, she joined the GOMICS lab at Westlake University as a postdoctoral researcher focusing on interference-based functional proteomics research. And last but not the least, we have Dr. Tianan Guo. He is the principal investigator at Westlake University. His current research focuses on developing proteomic technologies for precision medicine, exploring diagnostic biomarkers for thyroid cancers, and understanding understudied proteins. Without further ado, let's welcome Drs. Chris Sander and Augustine Lunar to share their talks.
Okay, can you still hear us?
Yes.
Sound okay?
Yeah.
Okay, so I'm going to share my.
You can share on your laptop or on the big screen, whatever you prefer. Your laptop.
Okay, so I'm going to share on my laptop. First, I have to find it. Okay. So I should be sharing some slides.
Yeah.
Yes.
You can present them if you want.
And
Great.
okay. Everybody can see it. I think they can. Okay.
Yeah.
So I'm going to start. Okay, so the presentation that I'm going to be giving is entitled Drug Combinations and Adaptive Resistance Mechanisms from Proteomics and Pathway Analysis. Just in case you want to reach out to me because you see something interesting in the slides, there's my email address, and it's also going to be on all the slides. So just by very quick way of introduction, cancer is the second leading cause of death in the United States. So this is a statistic that came from before the COVID pandemic. And so one of the things that a lot of the work focuses in on for us here in the Sander group is thinking about better treatments for cancer. And for that, we need to understand more about the complexity of cancer biology. So one of the just a node paper where we talk about some of that is looking for biomarkers to chemotherapeutics in an article back in 2015. Some of the challenges to trying to figure out how individuals respond or different cells just respond to particular drugs is one of the things is that we're challenged by is limited data. Another one of the big challenges is just the complex relationships that exist between DNA, RNA, proteins. Just a statistic that maybe for the proteomic people they might already be familiar with, but it's still kind of interesting to see is there are low correlations between transcriptomics information and proteomics information, somewhere between 0.3 and 0.6. A recent paper that we actually did with Tianan is one where we were looking at the predictive power of proteomics for drug response in comparison to mutations and also gene expression back in a few years ago. Another big problem that we're faced with is algorithmic shortcomings. So this is basically where the algorithms that we tend to use, many bioinformatics tend to use, they don't really understand about biology. These are algorithms that maybe just like a simple linear regression or more complicated methods, but still that they don't understand biology. They have they're not grounded in biology. Lastly, and kind of the big main topic that we'll be discussing here is how single therapies can it can be ineffective, or where the inefficacies arise from single therapies. And part of this is around drug resistance. So that's going to be the main focus of the presentation. So next, Before I get into the main method I'm going to be talking about, which is target score, I'm going to talk a little bit about the ecosystem pathway-based methods that have been developed in the Sander group and I've been a part of. So here on this on this slide, just a quick overview of a lot of the different tools that we have in for an ecosystem that I'm calling that we call Pathway Commons. So we've got kind of a range of tooling all the way from being able to create pathway diagrams where users want to kind of share their ideas as a pathway representation, to be able to search through all of the data that we have in Pathway Commons that I'll get into a bit on the next slide. And so we also have various tools and a lot of different languages. Also mapping, so you might have experimental data, you want to map it onto pathways. So we've got tools for that. And we also have a number of tools for actual analysis of pathway information along with experimental data. So in the red box there, I'll be talking about target score and Chris will be talking about perturbation biology and something known as cell box. So just a little bit more information about Pathway Commons. So Pathway Commons, it's this rich resource for pathway molecular information. And you can think of various scenarios where pathway information becomes very important. So it could be interpreting omics information, thinking about treatment strategies for patients. And so in Pathway Commons, we actually integrate 22 different databases. So we standardize the information for 22 databases of molecular interactions. We have over 2 million interactions covering thousands of processes. It's tripled in size in the past decade. And a more recent effort is how to expand beyond this. And we're going basically straight to the source, to authors, and trying to get them to contribute to the Pathway Commons resource via a project called Biofactoid. And there it's mutually beneficial in the sense we are able to make more use of the information and authors are able to share their content with a wider audience in a structured way. Also, one thing about Pathway Commons information, how it's different from things like StringDB or other databases you might be familiar with, is Pathway Commons information has interactions that go beyond the simple binary interactions of A interacts with B. We have interactions that tell you, for instance, that protein A binds to protein B to form some complex C that catalyzes some reaction D. As I mentioned, we've got libraries in various languages, and we also have some online tools that people can use. Okay, so now the heart of the presentation, I'm going to be talking about target score and how we use. Yes.
Sorry, can I have a question for the previous slides?
Okay.
So A binds B to form a complex C to catalyze reaction D. Is this, do you mean only direct interaction? Pathways may be functional consequences, not only direct interaction.
Yeah, so we're more focused there on direct interactions as opposed to more genetic interactions that are indirect. Yeah, so literally one protein binding to another to form a complex, direct interactions, yes.
Okay, thank you.
Okay, so going ahead, so target score for trying to mitigate and find strategies to deal with adaptive responses that occur in disease, especially cancer. So this slide, I think, illustrates the point of the necessity for dealing with drug resistance in cancer. So we've got a lot, there exists a lot of different inhibitors of proteins, cancer proteins. And some of these inhibitor drugs can elicit some very dramatic responses. So what you're seeing on the figure over here on the right is a drug that is super well known. It's in cancer, it's called vemurafenib. It targets a very specific type of protein and alteration on that protein called the BRAF V600E alteration. So in the first panel here, we've got baseline, a cancer patient. And you can see all the black dots being tumor in the body. And so this is at baseline before drug is given. 2 weeks after the treatment here, you can see the black spots have reduced greatly, and that is all a benefit from vemurafenib, which is great. But the problem is after 6 months, these metastases return, and the drug is no longer effective. So I think this really illustrates the necessity for dealing with drug resistance. Okay, so now I'm going to talk a little bit about the methodology of the target score method. On this slide here, on the left-hand side, what you are seeing is kind of just to try to illustrate the idea of where resistance arises in cancer. So in the yellow node here, we've got some drug, and that drug has, it is an inhibitor for certain biological entities, certain pathways, and it's able to inhibit those. And that might turn down the effect of downstream effectors, and it might elicit a therapeutic response, as you saw in the previous slide. And if that's where things ended, that would be great. That's what we all desire from drugs, cancer treatments. But the problem is that there is the potential, if there is these downstream effectors, they might be affected in such a way that actually turns on upstream regulators of the pathway that you were originally targeting. And if these are no longer regulated, they might in fact turn on parallel pathways that then lead to an adaptive response. And this is what we don't want, what's undesired. On the right-hand side, so in a second, I'm going to show some of the equations related to target score, but just to try to give you a general idea, overview of what is going on here. So with target score, what we've been using it with is data that is measured, proteomic values that have been measured after a single drug perturbation, and we calculate for each one of the proteins that was measured in a system, we calculate a target score for that protein. We have the first part of it is of the equation around target score is a function score. And so the function score can give you an idea of whether or not a protein is an oncogene. And so this is where it is positively affecting the growth of a tumor, or a tumor suppressor that is suppressing the growth of a tumor. And the second part of the main idea of target score is thinking about how proteins are changing by themselves within a system after a drug perturbation, and also thinking about the network change around the neighborhood of an individual protein. So as far as
sorry.
Yes.
So again, the neighborhood change, do you mean physical interaction, or can be indirect interaction?
So we might mainly think about physical interactions, but you'll see in a way to get some of the network. Ask me, okay, let me continue, and then ask me that question again if it's not clear on the next slide. But okay, but just to give you a sense of the idea. So what we're interested in is targeting adaptive responses with target score, trying to find ways to mitigate adaptive responses. And so with here in this formula, what would be an adaptive response, a positive target score, is oncogene that would give get a positive function score, and an increase in this combined change of the self plus the network change. That is what we would consider an adaptive response, as opposed to tumor suppressor. That is, we want tumor suppressors to increase in their abundance to incur a therapeutic response. So here is the kind of the workflow, the math around the target score method. So in the first thing, we've got drugs. So I'm going to talk a little bit more about JQ1 in a second, but we give a set of samples that includes all the way from sensitive to resistant samples, the drug, and then we measure proteomics. And in the case of what you'll be seeing in the presentation, it's RPPA, antibody-based proteomics. And so going to Tianan's question about exactly what the network is. So we actually combine in this setup, we combine data that comes from curated databases. And this, as I was mentioning before in Pathway Commons, it's more direct interactions. But then also to try to give context to the network. So trying to find a network that is specific to breast cancer, and I'm going to be showing examples related to breast cancer. We take information from another system, a breast cancer, I'm going to show it in a second, but breast cancer data that came from breast cancer patients, and we use partial correlation to infer a kind of a reference network. And this starts blurring the line of indirect versus direct, because here it's partial correlation based. So that can bring in some indirect interactions. And hopefully that answers Tianan's question. Point 3 here. So this is kind of just some of the math. So the function score, it can either be a +1, a -1, or a 0 if we don't have a clear-cut definition of whether a protein is oncogene or a tumor suppressor. We've got the network model that is coming from step 2, and we've got target score that is basically now the math, but the idea that I was representing on the previous slide. So we calculate target scores for each one of the nodes in the network, and we come up with target scores for all of the entities that were measured in a particular system. And then we start looking for modules of high target scores to try to see if we can kind of get an idea of what biological processes might be affected. Then in the last point here, we move on to dealing with experimental validation if we were able to find something that complements the original drug. This code is available on GitHub as an R package. And right now, the tool paper, we've got it on bioRxiv, but it's in review. So a little bit about how the network is derived. So we're actually using an algorithm called prior G-lasso. And so G-lasso is an algorithm that other people developed, but we are adding this prior information into it that comes, again, from the Pathway Commons resource. And as I was mentioning, we are taking data from patient samples, 900 breast cancer patients, and that was collected as RPPA data. We run it through prior G-lasso, and we get a network. Here is just kind of a very quick representation of the reference network, and I'm showing it in this particular visualization to show you that there are distinct biological processes that are visible within the network. So for instance, the first module in the light blue is a module that is around the activity related to an important breast cancer protein called ERBB2. Another thing that we did kind of in the validation of the reference network is to think about what happens if we reduce the original input information to see if we can retain the edges that we see in our final reference network shown in B. And for the most part, we do see, even at 50% data, we see 80% of the interactions remain. Okay, so now I'm going to get into a bit of the application of target score.
Augustine,
yes.
I have a question.
Yes.
The previous slides. So you mean you mentioned perturbation biology, but there are TCGA tissues. How did you perturb them? Or do you consider
no. So this is only to, so what was perturbed is in step 1. This is where the perturbations come from. Yes. And the reference network, this is from the TCGA. So this is a static network that is just kind of used as the background model for which we run target score over. Does that clarify?
But the perturbation is cell line. See?
Yes, perturbation of cell line and your.
Sorry, we cannot hear you now. Somehow we cannot hear you. Can you hear me? No, still cannot hear you.
His phone is closed, so I think he cannot
Your
speak.
phone, your phone is muted somehow.
Yeah, can you open your phone?
The microphone is
Dr.
muted.
Sander? Dr. Sander?
Hello, Augustine. Okay. So, dear Augustine, can you open your phone?
I
don't see any. Can you hear us?
Yeah, now yes. Some echo background.
You're not unmuted, are you? No, you're
Ah,
unmuted.
yes.
Good. Okay. Can you see things?
Yes, I'm not in the full screen.
Yeah. That's updating.
Oh, it's on yours. That's great.
Hi, dear Augustine. So, may I ask one question? Dear
Sure.
Augustine? So I think that maybe you have the data of the participation by one drug. So you can find that the components of a network will be changed. How you can predict the effects of multiple drugs? And could you tell me where is your training data set?
Yeah. So okay. First, just to get the slides in the right place, can all of you see the presentation still on slide 13?
Oh, this is what they can see.
It's 11. It's 11.
Here I
Okay.
see 11 years.
Because my screen sharing is paused. Why is my screen sharing? Oh.
Let's go sharing fast.
Maybe you can stop and reshare it.
Well,
Yeah, I'll share again.
No.
Slide 11.
Okay.
Well, we can go here if you want.
Yeah, but you can hear me, right?
Yeah.
Okay, we're gonna switch to the other computer that we have here and share the slides. So I'm gonna stop sharing. Sorry.
Okay.
Okay, can you see the slides?
It's loading the first slide. Yeah, the front page.
Okay, so okay, the question was, how do we predict how do we predict the effects of a second drug combination? And so for actual prediction of how a drug combination might work, you should wait for Chris's presentation. So this is kind of a more simplified method than the one that Chris is going to present, which is Cellbox. And so here it's less about predicting how a drug combination is going to work, and instead it is about identifying simply just through a calculation, an equation, trying to identify what is going up or down, and then letting the researcher kind of given a choice of what they might know outside of the analysis and the calculation what we're doing within the neighborhood, to give them options of things that they might want to, that they are seeing as going up or down, and then using their knowledge to select from that and then picking from that. So let me go through this example of a paper that we recently published and came out last year, where we're actually using target score. So JQ1, and it was the drug that I mentioned before, it is this drug that affects indirectly the MYC oncogene. And so it does that by preventing basically another protein, BRD4, from being recruited. And that kind of leads to a chain of events that affects the way that RNA polymerase II works. So what we did with this drug is that we tested, let me put a laser pointer so you can see. So what we did is that we tested a number of cell lines, going from cell lines like SCOV3 that is sensitive to the JQ1 drug, all the way to cell lines that were more resistant, HCC 1954, that were more resistant to the JQ1 drug. We then ran target score, and target score identified MCL1 as being a protein who its activity plus the neighborhood around the protein increased in a dramatic fashion, and that it might be something that that should be for consideration as a partner with the original JQ1 drug. So there's no specific prediction of the drug of a drug combination. What there is kind of just information on what is changing. So then in here on panel C, and just again thinking about whether or not MCL1 is an important drug that we might want to consider, we started looking at, again, TCGA data and seeing that across all different forms of breast cancer, that there were significant copy number alterations, amplifications, and gains, and that this is a drug that potentially should be considered to complement its inhibition with the inhibition of JQ1, or using JQ1 to inhibit MYC. So here on this slide, what you're seeing is that follow-up to the just running target score is on the original set of experiments that tested 5 cell lines was then to actually test out a set of 10 cell lines. Some of them are the same ones from the previous slide, and some, like BT20, those are new cell lines that we added in, and just to see the response of JQ1 by itself, MCL1 by itself, and the combination of the 2 drugs. Here in panel B, what you're seeing is kind of a summary. So wherever you see green, that is where there was a synergistic response, red antagonistic, yellow somewhere in the middle, additive. The next set of experiments that were done as part of this was then trying it out in mice. So here, either running a control or the single drugs or the combination in mice using xenografts derived from a cell line, this 468 cell line. And the combination was the one that reduced the tumor volume in these mice over a 4-week period. So that's kind of the end of the first story and the first paper and that we published last year. So we have a follow-up paper where we're kind of just going deeper into this resistant mechanisms around MCL1. So what happens if we then produce a resistant cell line that is resistant to MCL1 inhibition? So taking this this HCC 1954 cell line, which originally is sensitive to MCL1 inhibition, if we then after treatment by MCL1 inhibition over the course of 4 months, can we try to identify where the adaptation is coming from? And so here what you're seeing on the right-hand side is what the target score analysis led us to see. And so here I'm highlighting what we ended up kind of picking out of these values that we received. So I just want to mention, so with MCL1, it's kind of hard, it might be confusing as to why it shows up. So it turns out that MCL1, the drug that we were using for MCL1 inhibition actually stabilizes the protein, and that's why it pops up as having a high target score. And then, but two of the other drugs that we found interesting in this collection and that we believe that we could target was GAB2 and PARP. So PAR is a product of PARP activity, and so there's inhibitors for this. But GAB2, this is a little bit stranger. There's no direct inhibitors for GAB2. So here on the panel C, I'm showing how we actually went about targeting GAB2. So GAB2 is sitting between receptor tyrosine kinases and SHP2, which is upstream of RAS, RAF, MAC-ERK. And so since we can't directly inhibit GAB2, what we ended up choosing is to inhibit SHP2. And on this next slide, I'm showing kind of the results of the experiments. We've got dose response curves as I had on the previous slide, or with the previous kind of work. So in this 1954 cell line, the single drugs and also the combination. And so what you're seeing over here, this is kind of again a summary little heat map where you see green in this HCC 1954 cell line, where the cell lines are sensitive to MCL1 inhibition. We're seeing them also be synergistic with either PARP1 or SHP2 inhibition. And then when you've got a cell line, HCC 1954, that is resistant to MCL1 inhibition, it's actually possible to get synergistic effect simply using PARP and SHP2 inhibition. Okay, so then, so that's kind of where things stand. So we actually have this in bioRxiv, and we submitted it for review. And so some of the reviewer feedback that we've gotten is they want to see a lot more experimental validation. So that's ongoing for experimental validation with the SHP2 and the PARP1 inhibition. Also, kind of just an expansion of thinking about this method. We're thinking more about additional data sources. So right now, everything that you've seen has been with RPPA. We want to move this method more into mass spectrometry, and more specifically thinking about single cell proteomics. So you know, it was great to hear Tianan invite us, and yeah, we're looking for collaborations to try to move this methodology more into single cell proteomics, and also application outside of breast cancer. Algorithmically, thinking about if we move into more proteomic information, we need more comprehensive networks than the one we had with the RPPA from TCJ. So we're thinking about that, and we're also thinking about further ways of doing data integration to think about the suitability of the secondary targets, thinking about things, for instance, such as toxicity. If we give particular, if we inhibit particular additional proteins in a system, what might be the toxicity that arises from that? Summary, just very quickly, we developed this target score method to think about drug combinations. And we also, in the work, we started to infer mediators of MCL1 inhibition, also via CL2. I didn't talk about this, but it was also in this paper that we have ongoing. And thinking about the adaptive responses in both short-term and also long-term with respect to month stress. And we found synergy of PARP and SHP2 in relation to MCL1 inhibition in breast cancer. And for the sake of time, I'm just going to leave it there with the acknowledgements, and that is it. And I know I went.
Well, they can be in charge of the timing.
Yeah.
I think that, you know, the whole thing was supposed to be, I think, an hour and a half. That's why I changed the timing there. So we each have 40 minutes. And so if there's some questions now, that's of course fine. And then after that, I go, assuming that we can go until 9:30 your time.
Mm-hmm.
Okay. Yeah, go ahead.
Yeah, so if there's questions, I don't know if I hope to answer the question that I was asked previously. So this is less about prediction. The method that Chris will present, it uses much more data, but there's an actual prediction of what might be the response to a drug combination. Any questions?
So, dear Augustine, hello.
Yes.
So can you hear me?
Yeah.
Yes.
So my question is that whether you will try some artificial intelligence method to reach a higher accuracy of your algorithm.
Yeah. So yeah, so that was kind of the main point that I was trying to get at with on this very first slide with limited data. With limited data, right? So in some of these cases where we got patient information, you know, we're lucky to get 2 or 3 different patients, or just a handful of patients, right? Information for them, and
okay,
it's difficult, yeah. So so again, we do have, this method is meant to utilize less information because many clinicians just have less information available to them. But again, this is also part of the reason that we I was also mentioning that we want to try to move this more into the realm of single cell proteomics to actually get the level of data that we might be able to use with artificial intelligence.
Okay, so Augustine, so I'm not sure whether you are familiar with a type of artificial intelligence technology named few shot learning or small sample learning. So that means although your data set is very, very small, but you can still get very high accuracy with very limited data. So
yes, but those methods are usually dependent on having kind of a big background model, in my understanding of it, right? So, I mean, if we were talking about ChatGPT or GPT-3, for example, there's already a large language model that was trained on billions of trillions of data points, and then you just kind of fine tune it to get to that few shot or 0 shot learning.
Yes.
And so we still need that huge abundance of information to get the original model. So in other projects, we are trying to get to that. And again, this is one of the reasons where I'm particularly interested in single cell proteomics to actually get us to that original data set that might be large enough to do some of these things, as you're saying, with few shot or 0 shot learning.
Okay, so I have a question to Tianan. So I think, okay, so single cell proteomics. So think that we have a lot of data on the single cells. Tianan, so what do you think that we can analyze the communications between single cells? So do you have any thinkings on this problem?
Okay.
I can comment on that, but it's
okay.
You know, so it's a longer consideration. It's very interesting if you perturb cell lines and you have information before perturbation and after perturbation. If the readout is at the single cell level, and you have, say, for one perturbation condition in a pooled experiment, hundreds of cells, then in principle, you're amplifying your input-output data set by a factor of several hundred. That would be fantastic. And I'll tell you about some of the methods we use to use such large data sets. However, the difficulty is matching the conditions before and after perturbation. So the challenged, not solved, in a pooled experiment is to match the single cells before perturbation and after perturbation to see the change at the single cell level. And that's an ongoing challenge that everybody faces, unless you go away from the pooled experiments where each collection of cells has code like the sgRNA for CRISPR-I or CRISPR-Cut, If you go away from those pooled experiments, then you have to go to single well experiments, and you're back to something that have to do 96 well plates or 384 cell plates, and that's much more expensive in terms of experiments. So there are two ways to go, and that's a challenge for single cell perturbation biology.
Thank you very much. Thank you, Chris. I think this study is very, very impressive.
Thanks. Yeah, so I guess now we'll turn it over to Chris,
Okay.
so he can.
All right. So let's leave your microphone on, your speaker on, because that's what we're using right now, so you can hear as well, right?
Yeah.
The speaker, the microphone, we have two microphones on, it doesn't matter. Okay, so. Thing here, which is. I just have to leave the full screen on, but I want this also on.
Here.
Okay, let's do it this way. I'll have to spread. Is it
Sure.
screen shared? I'm not sharing the screen. Okay, I can share the desktop. I actually want to just share Microsoft. Let's see if that works. Yeah. Okay, and then I'll do this. Yeah. Okay, that should be okay. And let's see, this one here. Get the person in as well. Okay, great. 听不懂吗？ They're not responding. Okay, fine. So, but first of all, thanks for setting this up, and it's good to be have as a panelist Yu Xue, Roy Sun, and Tianan Guo. And please chime in and make comments. Tianan, of course, we've known for a long time when he was in Europe, and it was a pleasure to collaborate with him also on some publications and some work using proteomics. And it's just a pleasure, only in Zoom, maybe sometime in person, to be visiting Xihua Daxue.
Hello, can you hear me?
And
hello, can you hear me?
Were you able to hear me?
Yes.
Can you hear us?
You are?
Yes.
Okay. I can
Can
hear
you hear
you,
me?
yeah. Yeah.
Oh, yeah. Oh, just now my microphone has some problem. I asked a few questions, but okay, let's wait for your after your presentation. Oh, you have a Chinese name, Ke Sanda.
Ke Sanda, 对了. Yeah, this is a friend of mine gave it to me. Where I switch around Ke, even though it should be my first name, but we switched it. So Ke now is my, and then Sanda would be the next name.
Cool,
Sanda
cool.
is reaching out, so I'm reaching out. Sanda, like this kind of gesture.
I see.
Okay, so
Sanda means you reach out in 3 directions.
Exactly, and I'm only able to talk about 2 of those right now, but there's always a third one.
Okay,
I might tell you very briefly the third one in the end. Okay, so first of all, just to remind you that my group has created this tool for cancer genomics called the CBio Portal for Cancer Genomics, which has a large number of tumor samples with lots of profiling based on other people's work. But we put this together with an interface that allows you to look at the changes in cancer samples across 30 different cancer types. And this is ongoing. There are about 20,000 people a week that actually use this. And this is continuing now with 3 lead PIs, Nikki Schultz, who has a group in New York, Ethan Cerami, who has a group at Dana-Farber, and my group at Harvard Medical School, including Augustine. And we're going to continue and add to this whole genome sequencing for cancer samples, single cell profiles, which are often RNA-seq from cancer samples, more mass spec proteomics growing, like the kind of thing that Tianguo in his lab is doing, and then also patient similarity, where you want to compare different patients and see how similar are they with respect to their alterations, so you can do personalized therapy on them. And so this is background CBio portal for cancer genomics. Do use it. And I'm going to set my timer here so we don't run over. Okay. So what I want to talk about is primarily pre-division biology that can be used for combinatorial cancer therapy. Now, combinatorial cancer therapy, Augustine already talked about. What's that? You have a cancer treated with a drug that might be successful, like the RAF inhibitor that Augustine talked about, but then there's resistance that occurs, and those patients are not doing well. Now you want to have a second drug that is coming in, and you want to block the exits for the cancer cells so that you shut down the cancer. So typically that might require a second drug to overcome the resistance to the first drug. And by the way, I'll put this in the chat. There's a podcast, the podcast called is Ask Different, and I don't have it in the slide, but I'll put it in the chat for the Google. So you go to a podcast, you search for Ask Different, and there's a podcast with that where I explain a little bit about that. It's about a half-hour podcast, and I think I'm number 20 in that series called Ask Different. And so that's what's pre-division biology? Basically, this is a way of taking the biological system as a complicated system. And you want to know what's happening inside, not just know, but be able to do computational models of what's happening in that system. And the way to do it, you do many experiments of perturbation and response. And then based on that, you build a computational model of what is happening in that system. And that's perturbation biology. And it's a general field for doing large-scale experiments to derive computational models of cell biology. And so the idea of perturbation biology is inspired by my background in theoretical physics. It's perturbation physics. This is the Linear Hadron Collider in France and Switzerland. And you take a system which is the nuclear matter, you perturb it many millions of times, you look at the output, the scattering that comes out, you measure all that in detectors, and then you build a model based on that, and this is the standard model extended for particle physics. Now, interestingly, the experiments in biology are much less expensive than these physics experiments. The models, not mathematics here, but graphically, the models in physics are much simpler, the basic physics. And biology, the models, like the network that Augustine showed, are actually much more complicated, because biological molecules have an individuality, and biological beings have an individuality that is very diverse and totally amazing, and that's how biology and physics are different. But taking this inspiration, perturb the system, measure the output, infer a model, and then use that model to predict something. That's the mantra, that's the procedure that we want to carry over to biology. So let's take perturbation biology with the same kind of steps. And this is just a cartoon of a cellular system. This could be a single cell, collection of cells, could also be cell-cell communication. You perturb this in some way, many, many, many times. You observe it in some way for each perturbation experiment many, many, many times. And then if you have enough perturbation and response, you can build a computational model. What's a computational model? Well, there are all kinds of computational models. Some of them can be graphed out as a network, but the key thing is to have mathematics, of course, that represents this. You could use machine learning for that, and happy to talk about some of the methods that could be used for that to represent the intermediate states, the cellular processes, by some kind of latent space in machine learning. Here, I will show you a way of making a model of cell biology using differential equations that are solved in the parameter space using techniques which are adopted from machine learning. Once you have a good model, whatever it is, there are all kinds of things you can do. First of all, you can figure out if you have a model of this to make a prediction. Take a perturbation, what's going to be the likely outcome, like Augustin showed with drugs. Or you can do what biologists call mechanism. You interpret what is going on the inside, the direct interactions, as Tianan asked, the indirect interactions, make an interpretation, a mechanistic interpretation, what's going on in that system, understanding it, quote unquote. Or if you want to have a desired output in terms of controlling the system, like in therapy, you then use this computational model to design a specific perturbation that perturbs the system in such a way that the output, which we also call phenotype, is as desired. So you want, for example, to have several drugs coming in. The system, if the model is correct, will respond, and then you will get stop proliferation of cells or redifferentiate them, which would be useful in cancer. But this could be applied to all kinds of areas of biology. And so this is not new, the idea of perturbing a system and then getting a model out. But in fact, it's been going on for 40 years or 50 years in molecular biology. But the old way of doing it is a very small scale. Here is, a reasonably experiment using gels to look at proteins or whatever. And these are experiments that take many, many months to do in the old days. Then there's a mental process, and then people write a paper, and on page 4 of their paper, they have some kind of a diagram, a pathway diagram that explains this data. But these are not quantitative typically, these are logical kind of diagrams, and often, of course, they're incomplete, even though a lot of discoveries have been made in molecular biology using this kind of small-scale experiments. Scaling it up a little bit, using not mass spec proteomics, but in this case, proteomics on chips. We use antibodies to inquire for each perturbation condition, what is the output in terms of a certain limited number of proteins for which you have antibodies, like Gordon Mills, when I was in Portland, Oregon, at OHSU, the RPPA method. It's reasonable because for each perturbation condition, you get several hundred proteins measured, but it's not as comprehensive as full mass spectrometry, where you can get at least 5,000 or 6,000 proteins. And then at this level, still, even with this data, you could make a model, as people have done, with some computation and some thinking that they typically draw out as a graph in their paper. Okay, now we want to take this to a completely computational way, and that's at the bottom here, which is the new way. This is from RPPA, different proteins measured, in this case, only 150 using antibodies, and maybe 1,000 different perturbation conditions. That's pretty good. Different perturbation conditions. So you probe the system, you kick it, look at the response, and then we make a computational model that's completely data-driven. No biochemical parameters used. These WIJ in these equations are interactions between proteins and phenotypes, such as growth of the cells. And the variables are proteins and phenotype. The W's are the interaction between all of those. And so if you graph this mathematical model out, which is in differential equations, as a graphical diagram, then again, each node is a protein or a phenotype. The edges are the interactions, which in the equations are the WIJ. And then the advantage relative to the old way of doing it, which is just a graph for mental processing in the brain, you can now do computational processing using appropriate software and execute this system in such a way that you can now apply many, many, many different perturbations, single drugs or combinations of drugs, in order to test, given this model, and assuming that the model is correct, what a perturbation that could be quite complicated would do to those cells, and in a therapeutic sense, pick combinations of drugs that have the desired outcome, such as reducing proliferation. And that's the idea of perturbation biology for the cancer problem, using drug perturbations or CRISPR-I perturbations, for example, and using protein measurements, either using array-based methods with antibodies or using mass spectrometry, which we are very interested in to take also to a single spot.
Hi, Chris. Sorry, can I ask a question? The previous slides.
Oh.
So for the perturbation, there are several response data. One is based on longitudinal. You have different time points. Another is different dosage. We apply different dosage. The drug or chemical may bind to a few or more proteins. So do you have a different treatment to this two type of response data?
So good point. The best, so in these equations, the perturbations are P sub I, and in principle, this is actually time dependent. This equations, I'll show it again in another slide. So the best data set is you have a cellular system, say organoids, you perturb it with different concentrations of drugs or different levels of I knockdown. So as you said, different levels, number one. Number two, the ideal data set, you measure this as a function of time, because you want to have information that tells you about the behavior of the system as a function of time. So if you have data at different time points, then it's much more informative than just having a single perturbation, and after 3 days, you look what the cells are doing. However, in this example, we only have a single concentration for each of the perturbation. And secondly, we only have a single time point after 3 days. There are other data sets, but they're very rare for time series. We have one that we use in melanoma, but the other ones, so the challenge is to do data sets which are time dependent, and then we can do a much better job to find the parameters in the system of equations which describe the time behavior of the system.
Yeah, just a comment, if you can hear me. So just think of what I presented as being kind of out of necessity. It uses less data, right? And what Chris is presenting is kind of a much more thorough analysis, but it also requires much more data, right? So there's a continuation between the target score method that I'm presenting and what Chris is presenting that you should keep in mind.
That's right.
So yes, it would be great to have time series data, and it would be great to have much more concentration data, so that right in target score, I was using a network that was a reference network that came from inferring it separately from what I was using in target score to create the target score values, right? But here with Chris's method and what he's showing, that he's doing both things at once, right? Like he's inferring the network at the same time as trying to predict, right? And that just requires that you have a lot more parameters that need to be fitted, and you need more data.
That's right. And you know, at the end, I will say that, you know, point out that we're looking for collaborations, and it would be great to, of course, be able to collaborate with your laboratories to do experiments at a larger scale, especially using proteomics in the right kind of a system, so that we can, including time series, that would be fantastic. So that's something we could consider as a collaboration.
Yeah, great. So if we have limited resource, actually we have limited resource, do you prefer to measure more proteins or measure more cells if we have a single cell technology, or if you prefer more perturbations?
Well, I'm tempted to say more proteins and time series. And then the number of perturbations can be limited to save effort if you have limited resources, which we all do.
Okay. Okay.
But it's a design of experiment that's actually important. You raise a good point. Number of perturbations, number of cellular systems you use, and then time series. Time series would be absolutely amazing if we could do that. Even 3 or 4 time points would be good, which is only a factor of 3 or 4 more, but they have to be very carefully controlled in the system. I'd love to do organoids, time series with 100 or 200 perturbation conditions, and read out of 5,000 to 6,000 proteins using mass spec. Ideally also phosphoproteins, but I know that in mass spec is a bit more tricky, and but it's doable.
Yeah.
So this is just a conceptual way, also called the systems biology, where the kind of things that I just described are done several times, you do experiments, you do models, and you do this several times until you have something that you think can be tested for a therapeutic application. And so just to explain the equations, they're time dependent. Xi are protein levels and phenotypes like growth. This is a linear differential equations where you sum over all the interactions. The Wij are the interactions in the system here. And those you have to find based on data, the interaction strengths. The perturbation is put in to reflect the experiment, and it can also be put in simulation to see what a new perturbation would do. And this is an empirical term that makes sure that the system, which is incomplete, stays within bounds and doesn't go either too high or too low in its reactions. This is like a mean field approach in a way, a very simple one. When you have incomplete data and an incomplete model, which is always the case, to make sure that the system, the computational system is stable. And so your task based on large data sets is to get these parameters Wij, just from data, no biochemistry. And if you do that, then you can do something that's quite remarkable. You have the system which you've perturbed. Again, these circles are proteins. You've perturbed them maybe using drugs or CRISPR-I on the smallish number of conditions. You observe the reaction as large as possible. These blue ones could be 5,000 or 6,000 proteins from mass spec. And then you build a model with enough data that then has the edges, the interactions between all of these. But now, crucially, you have the interactions between the things you have perturbed, but also between all the things that you've observed, but never perturbed. And then the system can be simulated computationally such that a new perturbation and any perturbation can be applied anywhere computationally, not just on the targets which you have perturbed experimentally, but all of the proteins which you have observed as a reaction to the system, because it tells the perturbation, the large number of perturbation experiments tell you how everything in the cell is reacting, everything that you've measured. So then you could infer computationally what would happen if any particular one or combinations thereof are perturbed computationally. And that is something that you could not ever do in the laboratory, because it would be thousands and thousands of thousands, potentially millions of perturbations, singles and combinations. And that's the power of the computational models, if the data is rich enough to give you these Wij parameters. And so why is it a hard problem? Well, here's an interaction. For a system that has only 25, not 5,000, but only 25 proteins interacting. And the solution space where there is an interaction or not with any particular proteins is very, very large. Imagine you just want to know whether there is an interaction and edge or not, which is a 1-bit question, 1 or 0, these black squares. And what's the number of possible solutions? The number of possible solutions, these Wij, are 1 bit for each of these fields in the square. They're n squared here. And 2 to the n squared is the total number of solutions. For 25, not 5,000, that's a number that is larger than the size of the universe. And this is, of course, a straw person that you want to kick down, but that's the challenge. So you have to use clever methods to get these parameters, these blue edges, these Wij, from large data sets, but they're still limited, not infinite. But there are clever ways to do that, to get to solutions which actually then represent the data well and can be achieved in reasonably finite time. Hence, the perturbation biology approach using different methodology, in particular to get these Wij by a minimization optimization procedure using techniques for machine learning, but not necessarily machine learning as such, even though that could be done as well. So once you have from data these interactions, which are not just 1 or 0, but they have a direction and they can have a strength, then you can execute these quantitatively to predict what would be the result of perturbing any of these singles or perturbations on effect on different protein levels, effect on phosphorylations if you have kinases, and importantly in cancer, effect on proliferation of cancer cells. And when you then simulate computationally You actually get a time sequence to compare it to the experiment I've described. You want to compare the prediction, which is the steady state solution to these equations, to the observation, which is the cellular system observed in our case after 3 days. But this should ideally be a time series, in which case you compare the simulation here to the data at different time points. And that would be, of course, be more powerful. And so here's an example, a smallish set of experiments where we use different doses, in fact, and different targets with using different drugs. And this is the perturbation diagram, which is the response of a particular protein to a particular drug in the cellular system. Red is up and blue is down, where this is a log ratio. And log ratio is a much better term than fold change, which is often used, because if you say fold change, you never know what changes. The log ratio of protein expression is a much better word to use, but that's just a linguistic comment. So you use that, and now here's one solution to get these parameters Wij. Again, you perturb, and now you minimize a function written down here, which is a loss function, which is the differences between observation and prediction. And this is a loss function. You want to minimize this as much as possible, the difference between observation and prediction. And you want to keep things as simple as possible. So it's a regularization term that says, don't give me too many non-zero parameters, like the G lasso that Augustine was talking about. And you do this iteratively quite a number of times, because you have to have different predictions and get closer and closer in the iteration process to the experimental observation. And in the end, if you do it right, and here we use the Atom optimizer from TensorFlow, which is a package for machine learning, but we're using just the optimizer, but it's a very, very clever optimizer, and to get these parameters in a way that solves the complexity of having too many solutions to go through. And that works. One technical advantage is this: if we use a TensorFlow and the Atom optimizer, we can feed the differential equations. Here they are, the xdt, here's the tangent, here's the matrix multiplication with the Wij, here's the perturbation term, here's the restoration term. This is now the differential equation written in what? Written in the computer code, but the computer code is such that the derivatives here in this differential equation are done not numerically, but by a computer program that does automated differentiation. That's different from numerical differentiation. It actually does differentiation of computer code. I think this is a very interesting technique. It doesn't always work, but for certain problems it works, and it's one of the inventions that has come out of machine learning and is implemented in packages such as TensorFlow or PyTorch, etc. And we adapted this from our friends, and then we get the Wij parameters by minimizing this loss function over many, many, many experiments. Again, the idea is you have to have enough experimental data to be able to be successful in getting the parameters by minimizing the difference between experiment and prediction. And now these are details that are not important to go through of how you train this system. This is the technical aspect of how you run this. In particular, you want to have plot out for the protein level that you measure, what's the difference between prediction, which is solving the differential equations, and experiment, which is protein measurements. Okay, just Dang Yixia. Okay. Be well.
Thank you.
Okay. All right. So and happy, of course, to share this. There's also publication where this is in detail. In the end, you want to have a system of differential equations that you can simulate successfully. And there, the criterion for success is whether or not the drug combination that you simulate in the system actually will work experimentally in the laboratory. However, to convince your friends, the other biologists, that this is correct, you also want to compare the interactions which you've inferred to known pathway knowledge, the kind of things that Augusta and others have done, pathway commons and other interaction databases, where people laboriously over much time have put into knowledge bases a set of interactions, direct or indirect, to refer back to your question, Tianan, Sometimes they could be indirect, but they could also be direct interactions. And this is always put into evidence in databases such as Pathway Commons or Gene Mania or StringDB. You want to compare the computational interactions, say between AKT phosphorylated on 308 and STAT6 phosphorylated on tyrosine 641. That's an interaction. Well, that's a negative interaction here. This is a statistical diagram of the interaction. Zero is in the middle, negative is to the left, positive to the right. So you can compare several runs of the system, which is a distribution to background knowledge in the databases such as Pathway Commons, which often is numerical, but at least it tells you whether the interaction is, what the direction is, etc. So it turns out that some of these computationally inferred interactions agree quite well with what is known in Pathway Commons and other databases, but some of them don't agree. Then the question is who's right and who's wrong, and it's difficult to know, because even something that's reported in publications, even in the fancy journals, which we all respect and sometimes hate because they reject our papers, and those may also be wrong, those interactions. The computation may be better, or the other way around. The data on which we use this haven't been rich enough, or the model solutions aren't good enough, or the differential equations aren't complicated enough. So the model may be inadequate. If in doubt, you say the model isn't good enough. But in the end, the key point, after you have a discussion with your friends in biology, the key point is whether or not you can actually simulate the system and then go through thousands of simulations, pick drug combinations, in this case, in melanoma cell lines. These are out of many thousands of computationally inferred drug combinations, which I'm not showing. Two that came out at the very top, and Augustin mentioned those. One is a RAF inhibitor that treats cells which is mutated in V600E, valine to glutamic acid in position 600 in that protein, and hitting c-MYC. But c-MYC is not directly targetable, so as Augustin explained, you use a drug called JQ1 that addresses the MYC axis. So both of these combinations come up high as predicting proliferation in effect in these cell lines, and then we just test it experimentally. This is the experimental data, and it shows, because this red area goes below the anti-diagonal in both cases. It actually shows that the effect is not just additive, but it's also synergistic, also shown in this curve where you have viability against drug concentration, single drugs here, combinations of the drugs at equivalent concentrations. So say 50% of one, 50% of the other is better than 100% of each individual, and that's called synergy. To have effective therapy in cancer, you don't necessarily need synergy. It's just interesting to know, because you know that you are hitting two different pathways in a way that actually is more than additive, and that tells you something about the pathway effects. One hypothesis that derives from that generalizes beyond this particular combination of RAF, MEK, and SMIC, is to think, what do you do to shut down cancer cells that might be resistant? And the idea is that the question is, the resistance that emerges, you don't necessarily know what the resistance is going to be, because you're treating initially cells which are known to be mutated in one particular axis. And so you want to cover upfront potential resistance mechanisms. The way to do that, you go downstream of the pathways where there are different potential pathways or known pathways that are converging on something that's close to DNA transcription. But you also want to hit the particular alteration in the particular cancer cells, in this case, melanoma mutated BRAF. So the idea is for the targeted inhibition to use something that you know something about. In this case, what's the mutation that's oncogenic in the cancer? But then to block the resistance, you try to go downstream, closer to where things converge onto transcription at the DNA level. And in this case, MYC is in fact such a candidate. So that combination at the appropriate concentration might be a good one. The problem, of course, is if you have too strong an inhibition of c-MYC, you would affect all kinds of other cells in the body. So you have to triage, adjust the concentrations in the right way for this kind of combination. And that's a challenge in developing cancer therapy. Of course, you want to go beyond cell lines, which we've used here, to organoids and mice and people, and hopefully you can do that efficiently. And the kind of things that Augustin is also working on. So the summary of this is that you have perturbation experiments, you measure something, you measure both molecules and what cells are doing in terms of growth, and then you minimize the difference between prediction and between observation and prediction. I already explained that, to get the parameters Wij. We call the cell box, which is in the process of being further developed, potentially also to do interactions in the latent space after doing encoding in the right way into the latent space using, for example, a variational autoencoder, but this is work in progress, and others have done similar things. If you have such a computational model, you can use it, as I said, not just to get a mechanism, but also to design control. But it all depends on having rich data and a good computational model. Otherwise, your design of therapy will not be successful. And you have to have control calculations to do that. The real challenge is to get more and more data. And here's what we're planning to do. Again, this is an area of potential collaboration. We want to have something like 200 perturbations, for example, using CRISPR-I, singles and doubles. And then we want to measure phenotypes in the right cell culture. Could be organoids, could be cell lines. We'd like to measure metabolites, certainly proteins on the order of 7,000. Phosphopeptides, if we can, would be more than 10,000. This is possible technically. To do this is quite an effort. But if we had this kind of data set of several hundred perturbations, then we could build network models that, instead of what I showed before, have 80 or 100 nodes, have the order of 500 nodes, carefully chosen initially, or later on up to 10,000 nodes, the entire collection of proteins in the cell, interactions between them. That's the challenge. But the challenge is not to have something very, very complicated. The challenge is to have something that actually works. So if you have either the smaller system with interaction edges and time dependence, or the larger system, the real challenge, the real criterion for success is whether or not the simulation gives you something that actually will work in predicting something non-trivial in cell biology in general, or in cancer in particular, where you want to shut down cancer cells. So we're now in the process of planning CRISPR-I experiments, not CRISPR-cut, and there are reasons for doing that. CRISPR inhibition, targeted using sgRNAs and using protein mass spec to measure proteins currently at the level of 5,000 to 6,000 in a pooled way, in single cell, gradually coming online. Realistically, one might get 1,000 proteins per cell. And Augustin will test these experiments soon with collaborators in a small scale pilot experiment. But to do single cell protein measurements for this kind of a purpose is still something that will be quite an effort to develop in the right way. And so this is what we want to do in this sense. You have to choose the right targets using CRISPR-I, for example, knock out or knock down the genes using these guided RNAs, and then carefully choose combinations, pairs of genes that we knock out CRISPR-I. This requires some choice of what are the best combinations to use, and using mass spectrometry to measure proteins, phosphoproteins, and I would very much like to also measure metabolites. We may also, and we had to do this in a collaboration with people at King's College, for example, and also at Oregon Health Sciences University, do imaging where, in addition to measuring protein profiles in cells, you take images that are properly stained to look at cellular phenotype beyond just whether they grow or proliferate, don't proliferate, but phenotype in different ways. What's the size of the cell? What's the migration of the cells? What's the morphology of the cells? This is work in progress. For that, machine learning that goes from the complicated images to a labeled statement of what the cellular phenotype is, maybe 10, 15 different descriptive cellular phenotype, that would be something that would be extremely useful. Again, that's an area of collaboration. This is our vision, if you like, or ambition for perturbation biology. It looks quite elaborate and complicated. The challenge to all of us, if in doubt, keep it simple. especially for the cancer problem, so we can get to solutions that can actually be used and move to clinical trials. And I will not describe all the different ways in which people have, of course, done perturbation experiments. Typically, what they do not have is large readout at the level of proteins. They do have a large readout at the level of single-cell RNA-seq. We have a paper which published together also with Augustine, where we have a survey of about 45 different perturbation experiments that use single-cell readout using RNA, single-cell RNA. And happy to share that. It's also on bioRxiv. But we do not yet have protein measurements for this kind of experiments, because that's difficult to do technically. So that's toward
Hey, Chris. Chris, sorry, can I ask a question here? So I noticed you didn't include RNA data in your study design. Can I have your comment on this? Why do you intentionally do not measure RNAs? Because that's much more cheaper and easier.
So we measure RNA, and in these experiments, our friends and we measure single-cell RNA-seq.
But for your experiment, you didn't mention RNA, the previous slides.
No, we didn't RNA because why? The most obvious reason is that drug targets are proteins. And we also know that the relationship between RNA levels, RNA expression, sometimes that's called expression, and protein expression is not a one-to-one linear relationship.
Yeah,
actually quite
Augustine mentioned that.
As you know. So we want to observe proteins because they're closer to what the mechanistically happens in cells, because proteins are the workers in the cell, and RNA for the most part are messenger RNAs. But if you have RNA-seq, and if you can derive from RNA-seq signatures, that is collections of RNAs that are characteristic for certain cellular processes, you could bypass the proteins to infer from single-cell RNA-seq what are the cells doing phenotypically. Are they growing or not, and what pathways are turned off? But it's tricky because there is not a one-to-one relationship, as you well know, between RNA levels and protein levels.
Yeah, cool. We have some ongoing, very large perturbation study. Maybe after this live streaming, we can have a closed-door discussion, a few minutes.
Yeah,
please
we can
go
have
ahead.
discussion. Right, and you know, of course, we're very happy as always to share our software or to collaborate with you by running it and so on. So let me just give one example, which is a smaller effort. This is much closer to target score, a method that's very similar to target score, which basically looks at the response after inhibition, in this case with the drug, ovarian cancer cell lines, and then looking at mass spec data. 5,000 to 6,000 proteins per perturbation, but this is a small number of perturbation, but rich readout using protein mass spec and Matthias Mann group with Papa and Kosha in Copenhagen, and then using something like target score to find out which proteins are going up, which are pro-proliferative, or which proteins are going down that are anti-proliferative, like pro-apoptotic. And when we do that using both controlled experiment, 5,000 proteins, or after inhibition with the PARP inhibitor, which is actually an approved therapy in ovarian cancer, and comparing control and taking the log ratio between the protein levels before and after, and using that to analyze the data in a way that Augustine described in target score, but this is done similarly. We then get a certain significance on the vertical axis of the observation, and you get negative or positive log ratio. These things are going down and these things are going up, proteins. And then we focus in the end on using a pathway analysis, similar to target score. And then one of the most interesting was this one here, lipid metabolism. And there are proteins involved in lipoprotein modification. And but to target any kind of lipid metabolism, which is actually very important in cancer stress and also in cancer proliferation, we take we analyze this in great detail, and several inhibitors. Let me just show one of them, which is fatty acid synthase, which just affects the entire lipid metabolism pathway by shutting down the synthesis of fatty acids in the first step, in many fatty acids, but the first step is palmitate. And then we take those inhibitors again, as before, the PARP inhibitor plus the inhibitor of the fatty acid synthesis, which is this one. Again, with the right concentration, 100% of one leads down to a 10 micromolar growth of inhibition roughly. And if you now use a combination, you get a more than additive effect, 50-50 versus 100-100 in terms of concentrations. And so this combination might work using fatty acid synthase, which is not a toxic drug, might work to treat ovarian cancer patients that have been treated with a PARP inhibitor, but after some time, the cancer has come back with a resistance mechanism. So either you treat them afterwards, after the resistance has arisen, or you do upfront perturbation with 2 drugs in order to prevent the emergence of resistance in the first place. This, however, is non-trivial to get into clinical applications. We've not done this in 5 or 6 different cell lines. We still have to do it in mouse models or organoids in between. And then we have to nominate this with the right partners for the clinical trials. And as you know, that is quite complicated and can be costly, but we want to push this into clinical applications with the right kind of clinical partners. So this becomes a general approach, not just for going back to perturbation biology, where the ambition is, and this may take several years, to actually get to much more richer computational model of cell biology that we can apply to cancer biology for target for combination therapy design, and also any other biological system. Actually, this would be quite general. I mean, the ambition of deriving a comprehensive computational method for cell biology within cells, between cells, and collection of cells would be, I hope and expect, to have large impact on different areas of cell biology. And that, I think, is an ambition that we alone cannot achieve. We can only achieve it in collaboration. And this is what I call network science, where we have to work globally and we have to be open in the communication, similar to what our discussion was at the Center for Life Sciences at Beijing Dashi and Tsinghua Dashi, where we in fact discussed that as external advisors to increase the bandwidth of collaboration between Chinese scientists, some of whom are just absolutely spectacular, and scientists in other countries who also have done some good work. And that will be the challenge, to go to a breakthrough in computational cell biology. And let me just end here and not talk about how we use our artificial intelligence to compute the risk of cancer with clinical records, and also not talk about how we just use protein design to design enzymes which degrade plastics, because we don't have enough time for that. Happy to do that another time. Thank you.
Thanks so much. Very interesting. Can I have a quick question? So I understand all your analysis, your models, tools are based on cells. But tumor is a bulk containing a very complex microenvironment. There are different cell types, and a lot of therapies now against the cancer are based on immune modulation. So how do you comment on that? Do you have any thoughts on, let's say, PD-1 treatment or combination? Yeah.
Similar to your thoughts. I mean, and we're working on that in collaboration with people. For example, one of our collaborators, Fabian Koscher with Matthias Mann, are doing spatial protein measurements, where they measure after laser dissection on the order of 300 cells, maybe 10 different cell types. Melanoma is an example in that paper, where they get at the heterogeneity for each of these regions, different cell types in this melanoma slice. They get 5,000 proteins measured, and they can show the differences between those different cell types. RNA-seq, of course, gives you another way, slightly less expensive. You can do RNA-seq in pooled fashion with sgRNA, and then you can distinguish it, from tumor samples, and then using clustering in single cell RNA in single cell RNA space, you get very interesting information about what the different subtypes are. It's important to then distinguish the different cell types, so you get the heterogeneity. We just have a preprint out that you can also look at in bioRxiv, where we actually have a computational simple method of testing how different are clusters of cells that describe heterogeneity, for example, a tumor sample. Now, coming to therapy, now you want to ideally attack these different cell types, because one obvious mechanism of resistance in cancer is that you shut down the majority of the tumor cells, but some minority of the cancer cells are growing up. They have not been as proliferative before, but then you shut down the majority, the minority grows up. And now, well, then you have to know what those are. And then you have a second drug for those, or some other strategy, which is alternating between one and the other. And this is a very important problem. You're spot on. One of the major problems in cancer biology is, in fact, the fact that treatment has targeted the bulk of the tumor and has not paid sufficient attention to the heterogeneity of the tumor cells and the immune cells in the cancer, which one wants to activate. And as you just mentioned, antibodies against CTLA-4, PD-1, and a few others actually will increase the T cell response by unblocking the blocks, by taking off the brakes. But the details are very important, because whether or not the T cells can actually are in the tumor tissue, can invade the tumor tissue, and whether or not macrophages through cytokines have been repurposed to actually aid the cancer in a pro-proliferative way. So this is quite complicated, and we have to get more information about heterogeneity of tumor cells, including the immune cells which are in those tumors, and then find the right way to address them. My answer, which may be simplistic, but I think may be correct, which is use combinations of drugs and antibodies, of course, whatever they are. Combinations, because the system is so complicated, you need to shut down the entire system without killing the host. I described this a bit in the Ask Different podcast.
Yeah, so I work with a lot of clinicians, and some of them raise a very critical question. So in clinic, it's very difficult to get tumor samples, tissues, but it's much easier to get blood samples or urine samples. So from blood urine, we can see some host response in response to the tumor growth, in response to the drug treatment. Do you have any idea? Let's say since we have very easy to get a longitudinal blood sample or urine sample, do you have any model, any thought to use this information, if we have this data, preterm data for the blood, over different time, different patients, to infer the tumor biology in the cell?
Yeah. So I mean, if you look in blood samples, of course, this is a tremendous challenge. And people have talked for quite a number of years now about measuring DNA in the blood by looking at mutations. For example, in pancreatic cancer has more than 90% mutations in KRAS at glycine 12 for the most part. And then they want to see the DNA in blood. However, there are other opportunities. In addition to getting mutations in DNA, you can look at exosomes, which people can purify from blood, and the small vesicles that contain interesting stuff. Not easy to do experimentally, but it's interesting as a potential. Or looking at protein spectra using mass spec. But as you know, getting protein from plasma has a tremendous issue of dynamic range, because some proteins are extremely abundant and the ones of interest are typically less abundant. Yes, there are ways of doing that, but for almost 2 decades, getting protein mass spec data down to low concentration proteins using mass spec has been a challenge, but it's moving forward. The one that I think currently is most promising is to take DNA from blood, cancer patients, but even before the cancer arises potentially, take DNA from blood and do DNA methylation, which can be done using sequencing methods quite inexpensively, but it's not done enough. DNA methylation, DNA in blood, once you take care of the healthy cells, but there's a minority of traces from the cancer. DNA methylation is A, characteristic of the tissue type, and B, when the cancer grows in the tissue, it also changes its behavior, it redifferentiates, and that can be seen at DNA methylation. There are companies that are doing this, but one cannot rely on the companies because they're behind closed doors, like the company called Grail, and it's also expensive. The method of doing DNA methylation for that purpose from blood samples doesn't have a patent attached, because that's not patentable. So that's something that anybody can do. I'm talking to people in Portland, Oregon. They want to do this in their own labs. You could do it at HuB Dashi, and definitely, I would at least hope you can discuss with your colleagues whether or not DNA methylation from blood can be established. We can talk more about it. I think it's a potentially a great way to look at tumor progression, potential recurrence, but even get the cancer before it arises. I think it's why just one more point. Why is DNA methylation interesting? I mean, this is not a comprehensive answer, but it's because DNA methylation, a mutation in KRAS, it's one sequencing trace to look at the variant. But DNA methylation, you can do low pass whole genome sequencing. This is, it's not expensive because, you go to Beijing Genomics and, in China, there's fantastic sequencing technology as well. And you can do low pass whole genome, get DNA methylation. It's not expensive if you know what you're doing. You have to have control experiments. And the signal to noise ratio for something that's distributed over the whole genome, the signal to noise ratio is much better than looking for that one mutation in KRAS or PI3 kinase that's characteristic of the tumor.
But how this can be linked to your model based on the protein network in a cancer cell? It must be relatively indirect.
Well, that would be, I mean, so I think the kind of perturbation experiments, I think protein readout is and metabolites are the ones that are most characteristic of cellular behavior. For DNA methylation, this is a short answer. My hope is we're also working, as I just explained, and there's also a preprint on that. We look at clinical records. I'd love to do this in China as well in collaboration. Clinical records of people's disease histories before they get cancer, and using artificial intelligence such as Transformer or GRUs to look at the time sequence of medical records to identify people who are at high risk for cancer. They don't have cancer yet, they're high risk, and then you put them into what we call a surveillance program.
That's early screening.
Program. And there you can do, for example, DNA methylation. You can also do imaging, MRI or something. Any hospital, a good hospital, and you have them as well, can do that. And that's my hope for DNA methylation, primarily to get the cancer as early as possible. But it's only affordable in a high-risk population. That's why we do a computation to identify in 1 million people, 1,000 people that are at high risk, and start there to do surveillance program.
Yeah, but I have a good news for you for the proteomics field. So initially the blood protein, there's a high abundance protein issue, but there are a number of ways to remove them or enrich the low abundance proteins. Right now we can relatively cheaply measure a couple of thousand proteins or even 3,000 to 4,000 proteins from a drop of blood, from blood. So this can reach a very cheap and a very deep plasma proteomics profiling, and for many samples. So this will provide. Sorry?
Are you able to do this in your lab already?
Yes. This is what we are doing. So so this is this is some recent progresses. So initially we didn't thought about this type of depth, but now it's realized.
That's fantastic. Is it easy to say what the main technical advances to get that?
Yeah, we use some particle to enrich the low abundance proteins, and then you can identify those low abundance proteins.
Okay, that's fantastic. That's very good news. Congratulations if you can do that.
So we talk a lot to the clinicians, and in clinic is much more easier to get blood samples at longitudinal data points. So this will be, but this is different from the cell biology because it's not from the tumor cell itself. It's secreted. It responds to tumor growth or drug treatment. So I think it probably need a new algorithm to use this type of data.
Yeah, this is fantastic, and this is exactly the problem that I talked about. You can use that first of all to look at recurrence of cancer, somebody who has had surgery, and then to see whether it comes back as a protein signature from plasma, from blood, or even in the surveillance program or screening program to get for high-risk people to get the cancer early. So it typically was not possible, but now if you've solved the technical problem, then it would be fantastic to get a signature, meaning a collection of proteins, the concentration of which is characteristic for early cancer. And if you do this in combination with the kind of things we're doing clinical records, then you would have a small population on which it's possible and on which you can afford to develop these blood tests. And that would be totally remarkable if you can do that.
Has your group produced any publications that are available with this, the blood samples?
So we have some publication of the COVID published in Cell Reports, but those are a couple of years ago. So this year we have an even newer technology. This is not yet published. But we can talk
about the COVID Pro paper. The COVID Pro paper used some of the proteomics on the blood samples.
Yeah, sure. The first cell paper is from the blood.
Okay.
So another issue is we also have a spatial proteomics technology. We use a tissue expansion. We can expand the tissue physically by 500 times in volume. And then you don't need a laser-assisted microdissection to cut out the region of interest. We can directly cut out the region under microscopy, normal microscopy. And then we can also compare different cell types, different region. This is also a new progress from our lab.
Can I ask a question about that? Is this similar to what the guy at Yale who developed this called expansion microscopy, but he only has a factor of 20. Are you saying you have a factor of 500 enlargement?
So one linearly is about 10 times.
10 times. Okay.
Yeah.
Okay.
But in volume, XYZ is about more than 100.
Yeah, he says, I mean, his numbers are between 10 and 20, and we actually have a collaboration with them. But it's if you use that, and you're saying you can use that, and you can then cut out the cells for protein mass spec?
Proteomics. Yeah. We can identify many thousand proteins, up to 5,000 proteins from the mouse brain in the model using.
And how many cells do you need to get 5,000 proteins?
Probably 1,000 cells.
Okay, but that's on the way to getting heterogeneity. Great, yeah. But isn't it quite delicate to do the from a microscope to cut out those cells? To use it using a laser dissection?
Yeah, that's expensive and not 100% success if you practice a lot.
Right.
So another progress in the field of proteomics is that we have a SAX-DIA. That means we can have a systematic approach to analyze the directly interacted protein complexes in high throughput. So just like you mentioned, a database is the pathway comments. They're using the protein directly physically interacting with each other. I believe those are from manually curated biochemical publications, probably a lot of work to get this data out. But nowadays, a systematic way, we can identify hundreds, thousands of complexes in different conditions. And my lab also, this is initially from Rudy's group, and we also use this technology here to study some of the system.
Do you have tagged proteins you pull down the complexes and you see whether the members of the complex aspect? No,
a different way. So basically it's unbiased. We last the cell using a very light detergent. Then the complex still remain in the solution. Then there's a chromatography called the size exclusion chromatography. So the protein complex of different molecular size, they will be separated. And we collect the fractions every few seconds or every few minutes, every 1 minute or half a minute. They collect a lot of samples. And you can construct the elution profile of the protein complex. This is an expensive experiment, but can unbiasedly characterize the protein complex in the sample.
But imagine you have an issue of low concentration, because in the chromatography, you only get a small amount. So you really have to have the mass spec be quite sensitive to get the proteins in those.
Yeah,
temperatures.
that's what we have now, and not 5 years ago.
Okay, that's fantastic too. That's
Mm.
great.
Yeah, so
Yeah,
now we
thank
have
you.
a panel discussion. Can we have the panelists online? Professor Yu Xue, already now 1 hour and 15 minutes, probably a relatively short panel discussion. Yu, do you have any questions, comments? And Ray?
So I think the study of perturbation biology is very, very impressive. From the theory, equation, simulation, and from the application. So too impressive. So my question is that, so whether you in your model you only consider the interacting partner of a perturbed node, but whether you are consider the secondary interacting partners in the network.
Can you repeat the last sentence? I didn't hear the words. The last
So
sentence. Do we consider what?
Yes. So whether in your model you will consider whether you will consider the secondary interacting partners, the interacting partners of interacting partners.
The secondary what?
Just you basically mean like indirect partners, right? Indirect interactions.
Yes.
Well,
Yeah.
so I mean, you say secondary. If you mean interactions, you know, A going to B to C, but there's also A to C, which is indirect. Right?
Yes. So what I think in your current model, the B will be considered. So what the C will also be considered in your model?
Yeah. So the answer is because that both are included in the model. Some of the edges, some of the connections, some of the WRJ are direct interactions. For example, enzyme substrate, a protein kinase, and the protein is phosphorylated. Some of them are indirect, where you may have 1 or 2 or 3 steps in between. And that, of course, is a subtlety and also a challenge, because you'd like to have all the direct interactions. But if with limited amount of data, that's impossible. However, for predictive purposes, it's okay to have A to C without having B in the middle, as long as the prediction from A to C is reasonably correct. And we've tested that in some cases. Also, another problem where one can actually clean up those indirect interactions using something called partial correlations, which we've used for another problem, like and for protein folding and also for other interaction networks. So the direct answer is yes. Some of those computer interactions are indirect, and however, for predictive purposes, as long as they give the correct downstream effect and the correct change in phenotype, that's permissible, provided that the prediction is accurate.
Another thing is that, for example, the efficiency, efficiencies, for example, the beta and so on, in your equations, whether this numbers will or variables will be changed in different conditions. For example,
Yes.
that you estimate some variables, but maybe these variables will be static or will be changed in different conditions.
Yeah, that's a very good point. So a major limitation of the way we do it is we have the cells growing in one particular condition. Even if you go to organoids, you would typically only measure them in one condition. One of the reasons why I want to measure metabolites is I want to also have data on the metabolic state of the cells, which is related to the condition which the cells are in. And but that's a clear limitation, that we cannot do experiments about all kinds of different conditions. However, the hope is, and this is just a hope, that the basic pathway interactions are robust with respect to external changes. If we get all of the interactions, ideally, those are used perhaps in different ways in different external conditions. but they would still be reasonably accurate. But it's currently a major limitation that the experiments are only done in one condition.
Okay. Yeah. Thank you
and
very much.
Also relates to the question about the few shot or the transfer learning, right? So, not every cancer acts in the same way, right? That's why cancer is such a challenging problem. So you can't just take data from melanoma and combine it with breast cancer and make a better model with the two data sets, right? So that's part of the challenge.
Yeah, thank you very much. Yeah, I know. Yeah, thank you very much.
Okay. Yeah.
So Ray, do you have any comments?
Just one sentence, if I may ask, Yushe. Sorry that I don't know you yet, but in 2 sentences, what's your expertise, if I may ask?
I'm working on the PTM, for example, phosphorylation, on the protein kinase that phosphorylate substrates. So I develop tools to predict some kinase, maybe even like the microarrays study, SCASite. I developed another tool named PTS. To just
Great. Yeah.
Mm.
Luke Cantley just has a new method out where he claims that for any peptide he can actually infer what the kinase is for the peptide is. You know that it's going on in talks.
Thank you.
Okay, I think we already spent almost 2 hours in this seminar. It's really informative, exciting. And actually we are doing a lot of perturbation biological experiment, but we didn't have this word, this perturbation biology. I know that now I know that this is from perturbation physics. And I have a lot of more questions. Actually, we have an ongoing, a very large perturbation biology experiment. We have already required over many thousands perturbed proteome data. I wanted to discuss with you, but I think today is too late. It's already 11 p.m. here. Maybe I would rather you email and we can discuss next time. Maybe have your thoughts.
And just keep in mind, kind of this, the difference in these various methods that were presented today. Some are meant for smaller amounts of data, some are for bigger amounts of data, depending on what's available.
It's great, and we enjoyed the 2 hours. It's great, and also meeting some new people. And then Tian'an, of course, it's good to see you again. And we can have another discussion where we maybe explore some direct collaboration. Just as you know, anything that we have in terms of methods and software, we can share the software with you. You can have some of the people in your group or at HuB to actually train some people to run the software and so on. We can also help them, and we can collaborate. The idea is whatever works best together to achieve a joint goal would be good. And also on the early detection of cancer. Perhaps if there's some people interested in looking at clinical records in Chinese hospitals, would require a direct collaboration, and then use your methods to actually detect the cancer early in blood, for example. That would be an amazing future collaboration. And if you have some people that want to work on that, we're very eager to collaborate with them. They can come visit us, or we can share the software. All of that is a possibility. Let's discuss another time if you would like.
Yeah. Thanks so much. Yeah, thanks so much, Professor Xue. And thank you, Augustine. Nice to meet you, and have a nice day.
Yes.
Yeah,
Okay.
拜拜。好，拜拜。谢谢。再见。